So my name is Matthew Bergens, and I will be introducing the class to the human T lymphotrophic virus today, or as it's more commonly referred to as HTLV. So before I get started, I want to provide a little background into the virology basics before I talk about what makes HTLV so fascinating. Remember that while there can be a debate as to whether viruses are considered dead or alive, all viruses are obligate intracellular parasites, which means that they they don't have their own metabolic activity, and they only have that inside the host cell. They might have either DNA or RNA. They have proteins, but they have no metabolic activity outside the host cell machinery. Um, Viruses are very small. The average virus is 1/100th of the size of a bacteria, so they're super tiny. They what makes them also fascinating is that they have either RNA or DNA genomes, and this is important because remembering back to the central dogma theory, DNA contains the information, and the DNA is then transcribed into RNA, and then it's translated into proteins. And proteins are the functional components of life. The proteins are what make everything happen. And because viruses can contain RNA, this makes them very interesting because they upend the central dogma theory in a way. And kind of the last thing that I want to review on is how viruses replicate. They replicate in one of two ways, or a combination of the two, either through the lytic cycle, and this is when a virus enters a cell, it hijacks the host cell machinery, it makes copies of itself, and then the cell lyses and it releases all the contents of the cell, including all the copies of the virus, free to affect other cells. And then the other major way a virus replicates is through the lysogenic cycle. And this is when the virus incorporates itself into the genome of the host cell. And then when the host cell replicates, it unknowingly ends up replicating the virus also, because the virus is essentially in the genome. And then at some point in the future, the virus can latently activate and re-enter the lytic cycle. So, the next thing that I want to talk about is in 1970, two researchers, Howard Temin and David Baltimore, they discovered the enzyme reverse transcriptase. And up until this point, we didn't think that we could go from RNA to DNA. It was DNA, RNA, proteins, the central dogma theory. And with reverse transcriptase, we were able to show that there are retroviruses and we can go backwards. But however, still in the 1970s, people believed that retroviruses, one, they couldn't infect humans, but then two, more importantly, that no viruses could cause cancer in humans. And then in 1980, a researcher by the name of Robert Gallo in his laboratory at the NCI, they discovered the first retrovirus when a patient presented from Africa with a blood cancer, and then when they isolated the PBMCs, the peripheral mononuclear the cells, they were able to isolate the HTLV1 retrovirus, and then they were able to show that there are in fact retroviruses that contain this reverse transcriptase enzyme, and these viruses can affect humans. And so by the 1980s, viruses were shown to cause around 15 to 20% of human cancers, and the first of many retroviruses were discovered, including HTLV1. And then around the same time, the HIV pandemic started, which was in 1981. And so I have here kind of the general retroviral strategy, and I think it's important to start here before we go into the specifics of HTLV1. All retroviruses have single-stranded RNA genomes. They have a host-derived envelope. They include reverse transcriptase, which it's an RNA-dependent DNA polymerase, and they all contain 3 genes. Now, some might contain more, but all retroviruses contain GAG, which is the antigen that is the antigen encoding gene. It contains POL, which is the polymerase, the reverse transcriptase, and then EMV, which is the different proteins that are expressed on the retroviral envelope. And again, the purpose is to incorporate the retroviral DNA into the host cell. And the cell will end up through the lysogenic cycle unknowingly replicating the virus and expressing the retroviral genes as long as with its own genes. And so to talk about our virus, the human T lymphotrophic virus, HTLV1, there's 4 subtypes. There is type 1, which is characterized by the disease, acute T-cell leukemia lymphoma. So this is the main disease that we see with people that are infected by HTLV1. Type 2 is a different leukemia, which is very rare. It's called CTCL. And then type 3 and 4 are very rare. We haven't been able to observe any specific diseases because we haven't really been able to see too many people with infection with HTLV3 and HTLV4. But HTLV1 is the most prevalent. It's most researched, and so this presentation will focus specifically on subtype 1. And so I want to transition and talk about the structure of HTLV1. So it has an envelope. It has surface proteins, transmembrane glycoproteins, envelope proteins, a whole sorts of proteins. It has the capsid, which contains a nucleocapsid, which in immature virions, it's a membrane that surrounds the inner viral core. But in the mature viron, it's actually wrapped around the linear single-strand RNA retroviral genome. It includes, the HTLV1 structure includes both reverse transcriptase, integrase, proteases in the immature viron, as well as the HTLV1 genome. This is, again, a single-stranded RNA virus, which is in the plus sense. It's linear and unspliced. It's also one long 8.5 kilobase strand, and it's 5' capped and polyadenylated, which, if we remember back to human mRNA is 5' capped and polyadenylated, so it resembles human mRNA. It has flanking long terminal repeats, as seen in the picture, and remember that once the virus penetrates the host cell, the envelope and capsid release all the contents into the cytoplasm of the host cell. And for HTLV1, one of the 6 non-structural accessory proteins is called TAX. And TAX recruits host transcription factors, it recruits other cellular proteases, and these cleave that one long single-stranded RNA genome into separate mRNA transcripts. There are 8 in the plus sense and 1 in the negative sense, the minus sense. But at the end of the day, what makes HTLV1 unique is, again, the retroviral strategy that it incorporates into the host cell genome and the host cell expresses the viral genes. And so now I want to switch gears to talk about the pathophysiology of HTLV1. There are 3 general ways that people are infected. The first is by sexual intercourse, and similar to HIV, herpes, and other sexually transmitted infections. The second major way is mother-to-child transmission, and this happens mainly through breastfeeding. And this is generally really unfortunate for the mother because the mother is mostly unaware of the infection and then unknowingly transmits the virus to the newborn. And so then that newborn is infected for the rest of its life. And then the third major way is through blood transfusion, blood products, sharing needles, or any other type of blood transmission. And all 3 of these are considered neo-infection. And in order for this infection to happen, there is a 3-part transmission that is included in this process. How the HTLV1 virus enters the cell is very unique. It requires 2 cells, a T cell, and this is the cell that becomes infected, as well as a dendritic cell, which this cell ends up helping the HTLV1 virus into the cell. There are 3 parts to this process, as seen in the diagram shown. there is the viral synapse, which I like to think of like the synapse between two neurons, which in this case is the small space between the T cell and the dendritic cell. Um, and this is where everything kind of happens. The second part of the transmission process is what's called a cytoplasmic conduit, and this is similar to like a tubule, and together with the P8 protein, the HTLV1 P8 protein, it promotes adhesion between the dendritic cell and the T cell, the CD4 T cell, and these help these cells stick together. And then the last part is the viral biofilm, and which is just tons of HTLV1 virons that end up coating the surface of the T cell. Important to note, though, is that the dendritic cells are actually rarely infected themselves, and the infected cell is the CD4 T cell. And then once the virus enters the cell, it embeds its RNA genome into the double-stranded DNA host cell genome through the reverse transcriptase enzyme. And then the virus can become dormant, and the T cell will just continue life as it knows it. It'll continue up mitotically dividing and being a part of the host immune system. But however, as the T cell mitotically divides, as it divides, it ends up replicating the HTLV1 virus, that's the genome that's in the membrane of the CD4 T cell. And this is what we refer to as clonal expansion. And what's important to note about this is that every progeny cell from that initial CD4 T cell that was infected has the virus in the same part of the genome, which is important, especially when we talk about cancer and the specific type of cancer that HTLV1 is associated with. But before we talk about the specific cancer that HTLV1 is associated with, ATLL, I want to provide some background knowledge into cancer. I've included two slides for what I think are honestly the best manuscripts that summarize the common characteristics between every cancer, and these are what's referred to as the hallmarks of cancer. And as seen in the slide, there are 10 hallmarks that the oncologists Hannon and Weinberg have described, but the chief takeaway, the principal takeaway that I want the class to take away from is that there are various cellular processes that go wrong when a cell becomes malignant. Cellular proliferation, growth, the things that both inhibit growth, whether cells will invade and metastasize, with how they'll stick together, how cells will replicate, whether cells will go into apoptosis, cell death, angiogenesis, and this is when the cancer induces the formation of new blood vessels to kind of sustain the rapid malignant growth. It can change the metabolic pathways that are used in a cell. It can send out signals to avoid the immune system completely. So there are many things that go wrong within cancer. It's not simply mutate and then you have a malignant cell. So cancer is not simple. It is a multi-step transformation. And in the context of our virus, HTLV1, the process is from an infected CD4 T cell to a malignant CD4 T cell. And this process is what we call oncogenesis. And then the other takeaway that is very important is that every cancer is different. A brain cancer is different from a skin cancer, which is different from a bone cancer and a thyroid cancer. Different cell tissues have inherently different cancers. And then additionally, what causes the cancer will also make the cancer different. In our case, a leukemia caused by our virus, HTLV1, will be different from the cancer that will exist caused by radiotherapy from a patient from a previous primary cancer, or a leukemia that results from an exposure to our carcinogen, like a benzene, for instance. And so in the context of HTLV1, the HTLV1 oncogenesis is characterized by principally two proteins. The first one is TAX, which we previously mentioned as the viral protein. And this protein is, again, a potent activator of early viral transcription. But once the virus is in the cell, it also has other important parts to it, where it is associated with the bypassing cell cycle checkpoints through mitosis, It prevents p53 from doing its job and the other tumor suppressors. And then as the cell divides, because these pathways are being bypassed, there's an accumulation of both genetic and epigenetic modifications, and that with enough mitotic divisions, the genome will become unstable, which is one of the hallmarks of cancer. And then the second protein that is associated with HTLV1 oncogenesis is HBZ. And this protein is associated with not only proliferation, but also inhibiting apoptosis. But what is important about HBZ is that it is implicated in the oncogenesis because it is found in every malignancy caused by HTLV. So every patient that has malignancy caused by HTLV1 has HBZ1 upregulated, and so therefore it's believed to have a role in driving the clonal expansion of malignant cells and the progression of the leukemia itself. And so the specific type of cancer, once we have malignancy that is being caused by HTLV1, we call this cancer adult T-cell leukemia lymphoma. There are 4 subtypes: smoldering, chronic, lymphoma, and acute. And the lymphoma and acute are the 2 more aggressive subtypes, and both these subtypes, unfortunately, have both very poor prognoses. There are many other clinical manifestations in patients once the malignancy has manifested. However, it's important to note that most people that are infected with HTLV1 are asymptomatic, and they just simply do not know they are infected until they are very sick. They go to the physician and find out that they have this adult T-cell leukemia lymphoma and HTLV1 infection. And then again, HTLV1 appears in the same spot in the genome of every malignant clonal cell. And adult T-cell leukemia lymphoma carries a lifetime risk of 2 to 7%, and the 7% is for males, and there's a lower lifetime risk in females. And then another major risk factor is a higher proviral load, which makes sense. The other major disease that is caused by HTLV1 is a myelopathy called HAM-TSP. And this has a very common differential diagnosis for multiple sclerosis, MS. It happens in about 1 to 6% of those infected with HTLV1. But unfortunately, it's just like MS. It's a progressively fatal disease, and most people end up dying within 2 years from the onset of the myelopathy. There are many other comorbidities of HTLV1 infection. But the one that I want to mention is that once you're infected with HIV, you have a much easier time getting infected with HTLV1, as well as the inverse. If you have HTLV1, you're much greater chance of being co-infected with HIV. But unfortunately for those infected with HTLV1, there is no vaccine. Just like HIV, we symptomatically treat the disease. We try to repress the viral load through antivirals. But the main difference between the two is that HIV rapidly mutates. So one of the challenges to an HIV vaccine is that by the time a vaccine is made to one strain, the virus would be completely different because it rapidly mutates. And so the vaccine would no longer be effective. But what's good about HTLV1 is that it has genetic stability, so the genome is stable across the strains. And so this is very promising for one day that we will, in fact, be able to create a vaccine for the HTLV1 virus. To treat ATLL, as with most other blood cancers, we treat with chemotherapy. And I've listed the major combination therapy that's used currently. It's vincristine, cyclophosphamide, doxorubicin, and prednisone. Sometimes we use stem cell transplants. Generally, they're allogenic. But I do want to mention that while the prognosis, the lifetime survival rate from diagnosis, it has increased, but there's still overall a poor CR, a poor complete remission from the disease. And then for the myelopathy, TSP-HAM, it's treated very similar to MS. It's symptomatically treated. Prednisone is used. It's an anti-inflammatory. It's a corticosteroid. And antivirals are also used to suppress the viral load. And this is mainly to try to suppress the viral load to prevent the progression of the CNS destruction. And then, the main fascinating theme with HTLV1 is that it's not globally distributed like most viruses. Granted, it is definitely worldwide, but it's not in every single country. But instead, it's clustered around the world, and even so, as far as one city it might have like a greater than 90% infection rate, and then the city right next to it will have a virtually no infection in the city, which is very fascinating. But again, because most people do not know they're infected, we have limited data globally, and we just can estimate. And so the estimated infection around the world is there's 50 to 20 million people that are infected worldwide. Japan has the highest, with greater than 1 million infected. This is the largest singular population in the world. But then there's pockets, as you can see from the screen, that Sub-Saharan Africa and the Caribbean islands, South America, these all have endemic populations that carry the majority of the burden. But as you can see, you might have one island that's infected that has a huge endemic population, but the island right next to it might not. But one of the things I do want to mention is that we do not know if the pockets are because the virus preferentially infects certain populations, or certain people, for whatever reason, are more susceptible to HTLV1 infection, or is it because we just have poor data to account for a true representation of the infected population worldwide. But because of this, I want to finish this presentation by examining the future of HTLV1 as of 2019, because most people in 2019 are still unaware of infection, and honestly, if you talk to most people, they've never heard of HTLV1. They've heard of HIV because it's garnered a lot of publicity, social support across the whole world, but most people are still unaware of HTLV1. And, we have ideas about the prevalence of it. The prevalence can be anywhere between 1.3 to 9.3 per 100,000. And again, remember, it's clustered in endemic regions. But we have very limited data sets that really give us the true morbidity and mortality. And then because HTLV1 is not as popular as other viruses like HIV or other diseases, other viruses and diseases, or other cancers, it has received a lot less funding, and in the recent decades, the amount of funding has been constantly declining. So I want to end this by actually an open letter I found that was published in 2018, so very recently, a year ago, by Gallo, who again was the initial researcher that discovered HTLV, the HTLV virus, back in 1980, excuse me. But he wrote this letter along with 59 other prominent researchers, doctors, virologists, very important people in the medical community across the whole world, and they wrote this open letter to the WHO. And what I want to say is that what they want to fight for is a more global effort into the research for HTLV1, to make it a standard test, so that way we can prevent because it's easily passed on from women to their babies. This is a completely preventable transmission. This is a completely preventable malignancy. And then again, with sexually transmitted diseases, again, it's easily preventable. So more research, more more support, more awareness is needed for HTLV1. And with that, I would thank you for listening to my presentation, and I hope you learned something about this relatively unknown virus. Thank you very much.